This video is for lab 6 equilibrium. In this experiment, we use two chemicals, KSCN, potassium thiocyanate, which is a source of thiocyanate ions, SCN negative, and the other one is ferric chloride, which is a source of ferric ions. So this is potassium thionate solution in the beaker, and this is ferric chloride solution. The first thing that you have to do is, so look at this potassium thionate solution. How does it appear? And when it says appear, in appearance, mostly you explain it with the color, colorless, yellow, pink, brown, and that goes here. So here you're going to write down how ferric ions, which is coming from ferric chloride, looks like, and how thiocyanate. And again, you answer that in terms of the color. Now, so what we do, we take, in step 2, we take a few drops of KSCN and a few drops of FeCl3 and we mix them. This is my step 2. And we mix them to form the product, which is FeSCN with 2 positive charge. It's a complex. So then you look at the color of this product and then you write down here. Now, in step 3, what we do, we take this our product and add 25 milliliter of distilled water and make the diluted product. So the color here is getting lighter. Why? Because we diluted it with 25 milliliter of water. So from this first page here, this is what you should be able to answer in table 1. Now what we are going to do, this is our step 4. The diluted product FeSCN2 plus, we put it in, we divide that solution into 3 test tubes. So all these 3 test tubes basically have that diluted product. Step 5, the test tube 1 here, this one, is still a control. It's the same test tube, this and this. The color looks a little bit different, but it's exact same. Test tube 1 is a control. Control means something that we will not change at all. Then what we do in test tube 2 here, we add a drop of potassium thiocyanate. This is one of our reactant. Now what we do, so you put a drop of KSCN and you swirl it, and this is what now your test tube 2 looks like. So here I added a drop of KSCN, swirled it, and now it looks like this. Now it says compare its color to the control. You're comparing its color with the control. Is it getting lighter or darker? And this caused the formation of more of what ions? How can you explain it? So basically your change 1 was that you added a drop of KSCN in the solution and in this one, and you saw the change in color. Again, change in color is lighter or darker. So in this case here, you should write down something like lighter or darker compared to the control. What was my stressor? Remember, this is my stressor because when I'm adding this, that's what bringing the change. Now in the last slide, I have an equation here. So basically what's happening, that when I'm mixing my KSCN with ferric chloride, I'm making my product. Basically SCN negative from here, Fe3 plus from here, making this product. And eventually this reaction goes into equilibrium. So when you have a test tube where with this solution into it, Basically, in each and every test tube, you have this chemical equilibrium reaction going on. Now in test tube 2, when you added a drop of KSCN, is that going to disturb the equilibrium? Yes. Now based on the color of the product, you will tell whether the reaction is moving to the left or right. That goes here, left or right. And if you're saying left, means you're making, so again, let's say this is your reaction. If you're saying it is going left, means you're making more reactants, then the reactant formulas go here. If you're saying you're making product, that means you are writing the formula of the product here. Now, then what we do in test tube 3 here, we add 1 drop of ferric chloride. Again, that's one of my reactants. And swirl it and then look at the change in color. Same thing. What is my stressor? I added ferric chloride, so that's my stressor here. Color here, lighter or darker. Now, so is it shifting it to the left or the right? Now you compare the colors. So if the color is getting darker, you have to think on which side the color is getting darker. If the color is getting darker on the product side, and by adding FeCl3, again the color is getting darker. That means you're saying it is shifting to the right. And if you're shifting to the right, let's say, that means you're writing the product formula. Now once you're done filling this table, and again you do not submit the table, you basically going to answer these questions under lab 6 quiz. The first 3 questions are based on change 1. The next 3 questions are based on change 2. And question number 3 and 4 are basically based on the lecture topic. And for example, here you're applying significance of K value, equilibrium constant. And here you are applying Le Chatelier's principle. And then you're going to answer these questions under the lab 4 quiz. So one, in general, I'm writing some general reaction, generic reaction. Let's say A plus B gives C. And let's say A is colorless and B is, for example, let's say pink. And when you're mixing these two, you see the color of the solution is changing to red. Then red will be considered as the color of the product. Now you have divided, let's say you diluted this C with water, and the color is, and then you are putting that solution in 3 test tubes. When you are diluting it, the color is going to get slightly lighter. And you have this solution in these 3 test tubes. First one is control. In the second test tube, let's say you're putting 1 drop of A. And the color gets darker. What does that mean? That means you're shifting your reaction to the product side, because on the product side you have a darker color. If the color gets lighter, that means you're shifting it to the left. If you're shifting it to the left, your species that increased are A and B. If it is equilibrium is shifting to the right, then species that increased is your product, which is C.